Hi and welcome. I'm trying to learn virology today, nature of the Viron, and after we've done that, we're going to do Tiptoe Yoga, and if you participate in the Tiptoe Yoga, then it's very honest. Nature of the Virons vary widely in size and shape. So even in size are the Virons changing widely. Viruses are smaller than cells, ranging in size from very small, 0.02 to 0.3 something, I guess nanometer. A common unit of measure for viruses is the nanometer, which is 1000 times smaller than 1, no it was not, whatever that is, and 1 million times smaller than 1 millimeter. Okay, 0.3 something. It must, no, I don't know. Okay, it's very small. We're going to find out later how small. Smallpox virus, one of the largest viruses, is about 200 nanometer in diameter. So 1 million times smaller than 1 millimeter times 200. So 50,000. 5,000. So a 5,000th of a millimeter is the smallpox virus, which is a very big one, so 5,000th of a millimeter. Poliovirus, one of the smallest, is about 28 nanometer in diameter, so 50,000th. Yes, 50,000. Well, 30,000 or something. 38,000. Something. As we have stated, some viruses contain RNA, others DNA, and the nucleic acid can be either double or single-stranded depending on the virus. Viruses can have either DNA or ribonucleic RNA acid as the genetic material, and it can be either single-stranded or double-stranded. Viruses are sometimes divided into two types based on whether they have DNA or RNA as the genetic material, and all viruses contain one or the other in the Viron. However, there's a third group of viruses that use both DNA and RNA as the genetic material, but at different stages of their reproductive cycle. Most bacterial genomes are between 1000 and 9000 kilobase pairs of DNA, with the smallest known being about 590 kilobase pairs. Interestingly, the bacteria with the smallest genomes are, like viruses, parasites that replicate in other cells. So the smallest bacteria are parasites, which are replicating in other cells. Sections 1622 and 1623 are going to talk about it. They are number 8 too. However, and we haven't made the 8. However, one of the largest known viral genomes, that of vaccinia, is only 190 kilobase pairs. Some viruses have genomes so small they contain less than 5 genes. Yes, but there are probably still these thousands of kilobase pairs. 5 genes. The size of a genome of a few representative types of viruses are given in table 8.1. So the H1 parvovirus has 5,000 bases. Why not kilobases? Okay, we know what we want to talk about. Some viruses have genomes so small they contain less than 5 genes. The size of the genomes of a few is, as can be seen in the table. The genome of some viruses, such as reovirus, is not present in a single molecule, but is segmented into more than one molecule. Yes, here we're having the Gopea mosaic virus, 2 different molecules. And the reovirus type 3 has 10 different molecules. The structures of virons, virus particles. Is that the difference? Virus particles. Virus particles, maybe everything can be a virus particle, are quite diverse. So the structure is differently, by some, varying widely in size, shape, and chemical composition. The nucleic acid of the Viron is always located within the particle, surrounded by a protein coat called the capsid. What if it's maybe sometimes the other way around, but it's just always classified like that, whatever the name for it is? What if there's some which has the nuclei at the outside, but no one recognizes it because everyone is saying the thing in the inside is the nucleic acid? Maybe it is possible just to check where is now the DNA or RNA or something, but just what if? And it has consequences to understand the virus if there's maybe someone making a wrong description where everything is. And if it's just done by definition, the outside thing is the other thing. Now, the nucleic acid of Viron is always located within the particle, surrounded by a protein coat called the capsid. So the nucleic acid inside and the protein coat called the capsid on the outside. We're having here the nucleic acid in the center and then the capsid around it. The terms coat, shell, and capsid are often used interchangeably to refer to this outer layer. And we're talking here about this outer layer, the brown ones, not about the envelope. The protein coat is always formed of a number of individual protein molecules called structural subunits. Okay, all these different virons have in the structural subunits, which are some types of proteins giving the coat around the nucleic acid. And that is arranged in a precise and highly repetitive pattern around the nucleic acid. The small genome size of most viruses restricts the number of different viral proteins. Okay, these proteins out of which the structural subunits, which is the coat around the nucleic acid, which is in the middle of such a viron, is limited in the diversity of kinds of proteins because they are so small. Some have only one. But most viruses have several chemical distinct kinds of structural subunits that are themselves associated in specific ways to form larger assemblies called morphological units or capsometers. A few viruses have only a single kind of protein in the capsid, so just one type of fence around the nucleic acid. But most viruses have several chemical distinct kinds of structural subunits that are themselves associated in specific ways to form larger assemblies called morphological units or capsomeres. Okay, some people have built a fence just out of one material, others are mixing. And that is then called morphological units or capsomere. It is the morphological unit that is seen with the electron microscope. If it's one who if it's too small and does not have multiple different types of proteins for it, and it is not therefore a morphological unit, can it then be seen with an electron microscope? The information for proper aggregation of the structural subunits into capsomeres is contained within the structure of a protein themselves. I don't know what aggregation means, but I think they're saying how it should be built is, does the protein now by itself? Maybe by the way it is built, the protein. But it can stick only together that way because it is built that way. If there are a bunch of sticks, they will not start to build on top of each other. They will fall down and start to build or lying down. No one will stand there. If it's big enough, then maybe one will stand. But all the first one, we will all, maybe they come like that, but we will all fall down. And that big heap of sticks, these sticks will all be in some way horizontal and not vertical. The information for proper aggregation of the structured subunits into capsomeres is contained within the structure of the proteins themselves. And the Overall process of assembly is what's called self-assembly. It could be differently, as I just said. Maybe there is a plan for it, how to be built, and they are reading themselves the plan and building themselves on that. But the other idea which I just had, which I just built with the way that it happens that way. For many viruses, I don't know which one is true. For many viruses, the self-assembly process is assisted by molecular chaperones, proteins that assist in folding and assembly, but that themselves are not a part of a final structure. Okay, they don't say how they do it, just that they do it themselves, so we don't really have to try to understand what they actually mean there. A single Viron generally has a large number of morphological units. Is it not one unit around one nucleic acid? Or nucleic acid is? The complete complex of nucleic acid and protein packaged in the virus particle is called the virus nucleocapsid. Okay, nucleic acid together with its coat, the sometimes morphological unit or capsomers is called nucleocapsid. Although the virus structure just described is frequently the total structure of a virus particle, okay, that could be all. A number of viruses have more complex structures. These viruses are enveloped viruses. Okay, what I just said, there exists also some which are just like that. So that was the first type, now I'm explaining the second type. And they have an envelope with them. These viruses are enveloped viruses in which the nucleocapsid is enclosed in a membrane. Viruses without membranes are sometimes called naked viruses. Virus membranes are generally lipid bilayer membranes, but associated with these membranes are often virus-specific proteins. Okay, it is a lipid bilayer, but with specific virus proteins. Inside the Viron are often one or more virus-specific enzymes. Okay, where are enzymes in? Do we say that here? We don't say. There are some type of enzymes in, and these are also maybe not virus-specific ones, but some which are differently by viruses as by other cells. Such enzymes usually play a role during the infection and replication process, as we will discuss later in this chapter. Okay. If you were here for virology, you can go directly to the next part of virology. Then we will continue with virology. If you stay here and participate in the tiptoe yoga, then it's very honest. We take together, breathing out the tiptoe, out and down, in and up, out and down, in and up, open to the side, in and up, open to the other side, in and up, open to the side, make an inch of space between your feet, in and up, out, sink down, in and up, out to the other side, in and up, out and down, in and up, out, lean onto the knees, breathe in, out, make a hip back, in middle, out the belly, in middle, out, come back around, back up at the top of the back, shoulder roll, arms down, heels, both heels and shoulders down, arms up, onto the heels, sink down, this can go onto the knees, in and up, out and never go down, and up, hands down, heels down, legs to the side, one foot points in front, the other one to the side, in, arms up, out, bend your front leg, cover the front leg on the tiptoe, in, reach in front, out and back, in, straighten the front leg, out and back, change the feet, in, out in front on the tiptoe, in, reach in front, out and back, in, straighten the front leg, out and down, feet a bit apart, pointing to the outside, in, the arms up on the tiptoe, out and down, in, and imagine to push something heavy upwards, out and down, in and up, out and down, in and up, hands down, heels down, feet together, in, up on the tiptoe, and out and down, breathe in, and stretch just the left leg and the right one, and change, in, middle and down, out with the feet to the side, breathe in, out, go over to one leg, in, out to the other one, in, out middle, close the feet, breathe in, and out, in, And out come slowly over around, back up at the top, backward shoulder. In the arms up, hands to touch and down to heart center. That was the Ditta Yoga. I thank you very much, and until next time.